Hello, everyone. This is Cassiano Carromeu. I am the Director of Research and Development at StemoniX. Today, I'm going to be talking about implementing iPS-derived 3D neurospheroids for fast-tracking novel target and hit identification in a human-first drug discovery paradigm. So it's a long title, but I think it's going to be very interesting. First, a disclaimer: I work at StemoniX. Our expertise in the human iPS-derived tissue, especially neurospheroids and organoids. We adapt these biology-rich platforms to high-throughput screening. And one particular that we're going to be talking today a lot is the neural platform that we have, or our experience with the neural platform that we call microbrain. So I'm not, of course, referring. I will be talking about a lot about our experience with that and how that could improve or help to replace or refine the use of animal in research. So I'm going to have two main parts on this presentation. The first part is going to be talking about this human neurospheroid platform, our experience that have acquired over the years. So introduce a human iPS-derived 3D screening platform and discuss the key aspects of this in vitro system that's able to reduce, replace, or refine animal use in preclinical research. And the part two, I'm going to use one example or a case study for a neurodevelopmental disorder, more specifically Rett syndrome. That's an autism-like disorder. It's using these iPS-derived neurospheroids from the disorder to demonstrate how this platform can be used for rapid target and hit identification validation, and finally provide an example of clinical translation using this screening platform. So you all are probably very familiar with the models that are available for preclinical research. So there's basically two main models. That's the in vitro model and the in vivo model. So usually the in vivo represents a little bit more complexity. They have complex physiology. They have multiple cell types interacting that also are very important, especially in some disease case. However, the in vitro model has several advantages, like there's a scalable, early, easy access, and less expensive than the in vivo model. The main problem is that they usually lack the complexity that you're going to require for the physiology for a complete organism, especially a human model. In the other case, in the other hand, they do have, because they're scalable, they do have the feasibility of being applied in a high throughput scale that usually is harder to apply with the in vivo model. Even animal models are very expensive to do at scale. And these models, these cellular models usually are a little bit cheaper. However, as anyone is familiar when you're talking about screening or research and preclinical research, is that there is no perfect model, right? So there's several different caveats of using these specific models. For example, the 2D cellular model, they do not recapitulate the complex human physiology. They're very simple models. Usually it's a one cell type only or a few cell type only. The most promising on this in vitro model, the organoid model, they suffer of a huge variability between the different spheroids, different organoids. The animal model, the ones that are going to be focused, do not fully recapitulate the human physiology. There's a lot of disorders or diseases that are really hard to mimic and to model using an animal model, and they're also very expensive. And finally, the brain here that represents the post-mortem tissue usually just represents a snapshot of that. They're really hard to get, and you are only getting what's available to you, and usually it's a snapshot of the disease or any point of the disease after the person died, for example. You can have access to the tissue. So they are usually for validation, but it's really hard to do a research, a preclinical research based on only post-mortem tissue. So one of the main caveats of the in vitro model is like how you now can have relevant tissue in vitro that can now help to drive preclinical research, right? And one of the big breakthroughs in the past was the discovery of the reprogramming to create induced pluripotent stem cells, right? So these cells are able to differentiate towards any tissue in the body. And because you with the reprogramming now, you can reprogram any cell to this pluripotent stage. Now we have access to a diverse genetic background, right? Basically, you can take cells from patients or fibroblasts from patients and use those fibroblasts to differentiate towards this tissue. So they are human-derived tissue. They are scalable because you can expand the cells, and you give it access to a lot of the diverse genetic background. So there is also finally several protocols available for this that's able to create a tissue pattern towards the tissue of interest, for example, neural tissue or cardiac tissue, and so on. There's two main models available, 2D models and the 3D models that refer are organoids. In this case here is showing the brain organoid that usually is a complex tissue, has multiple cell types, have the organization that you cannot mimic in 2D. One of the main caveats of the organoids, it is the fact that they're variable. So with that in mind, one of the things that's emerging is less variable 3D cultures, right? So and to be able to contribute for preclinical research, there's a lot of things that these 3D tissues need to have. One of the key features for this human iPSC-derived 3D cultures is that they first provide access to diverse genetic backgrounds, as I mentioned, like because you can reprogram iPS from patients, now you have access to a lot of disease lines. They are composed of co-culture of multiple cell types, and a lot of diseases, these are very important. For example, the example that's shown here is a cross-section of one spheroid, a neurospheroid, that's composed of neurons and astrocytes, cortical neurons and cortical astrocytes. That's really important for some cortical disorders. And the interaction between these cells is really important. You can even see in a high resolution how these cells interact in 3D, and you start to see that this 3D interaction, these cells are really close together, and the astrocytes and neurons are interacting in 3D in a way that more closely resemble what's normally found in the in vivo tissue. These are also active, right? So these need to be functional tissue. And one of the characteristics, going back to this neuronal tissue that we have it here, is that they present synchronized neuronal activity. And finally, because we're doing this from in vitro system coming from iPSCs, you can now scale this for platforms for high throughput or high content screening platforms. In this case, you could use like 3 to 4 wall plates, for example, and have this in a format that will allow you to screen several compounds at the same time. So you will have an advantage to the in vivo system that now in a small format, you are able to screen several compounds at the same time. So, in summary, this neurospheroids, the desirable in vitro model for a high throughput screening, they need first the correct cell types. In this case, as I mentioned, at least the cortical neurons and astrocytes. They need to have the correct gene expression, and they do express a lot of the key features or key neuronal transmitters that are the markers of neuronal tissue. And they need to have the correct functionality. In this case, neuronal tissue, they're electroactive cells. And what you can observe, what we observe this tissue that we have done in the lab is that they have the correct electrophysiology, in this case represented by MEA, that's multi-electrode array electrophysiology. But they also, you can also see this electrophysiology indirectly using calcium fluctuation. So you have calcium tracings that's able to have this burst that's shown here by calcium oscillations on Flipper. So, and the Flipper is just a calcium reader, a high throughput calcium reader. And what you saw is that these calcium oscillations represent the synchronized burst that this network is showing. That's is quite interesting from a neuronal standpoint. We are also able to modulate this activity. What's really important, because not the activity, the tissue need to be active, but need to be relevant. All right, you need to be correct the correct gene expression or neurotransmitters that are able to modulate that activity, and you can modulate this activity agonists or antagonists of specific neurotransmitters that are present on this tissue. And this is just showing the modulation of the calcium activity. So you're modulating the calcium waves or the calcium frequency of the peaks that you observe using this platform. One thing that if you really want to apply this on a large scale and to gather or to replace some of the animal use in preclinical research, is that these need to be reproducible over time. So not only batch to batch, but from experiment to experiment. So you need to find the same modulation that you find with the previous batch. And this is really important because high throughput screening platforms, to be classified as high throughput, you need to be able to be reproducible over time. And also you need to be able to manufacture this at scale, right? You need to be able to demand this to several research, several labs at the same time, or to your demand on the research that you have. If you cannot reproduce a very few, this may not be feasible for the kind of research that we're doing. So these are all the key features that these platforms need to have to be able to start to replace or to start to think about the use, concomitant use of animal in preclinical research. So just to summarize, they need to have relevant cell types to expect physiology, and they have a robust screening platform that need to have all these functional characteristics that I mentioned. So across the world, there are several labs using these platforms for different things, and there's different uses that can be done. We particularly have used some of these 3D models. So we have used these human iPS-derived models for genetic disorder studies. I'm going to show one example. For functional high-throughput screening with target ID and compound certification. This is only in the drug screening, drug discovery scenario. But you can also use for safety pharmacology or tox, for example, for early hazard identification for neurotoxicity or environment toxic screening. And finally, you can even use it for new disease modeling, for example. You can use these 3D models and integrate new cell types on this. For example, you could use these neurospheroids, that's basically a representation of the brain, and then integrate glioblastoma lines to study a cancer model on these cells. So just in summary for the part one, we take these human iPS-derived 3D models and they give you access to a lot of relevant biology in vitro. They are a scalable and reproducible platform for physiology interrogation. It is really important when we're talking about a high-throughput screening platform. And finally, they can better recapitulate the complexity of the human tissue if compared with the 2D. Of course, there is still a gap between what you can find in vivo, but at least these in vitro models are trying to close the gap between the in vivo and the in vitro. And this is really important. There are steps that's in the right direction, steps that's really important to go towards that. One thing that there's no perfect model, no perfect screening model for everything. There is the caveats of the in vivo, there's the caveats of the in vitro, but if you have the right questions for that model, you can have very powerful answers. And some of these questions can be already answered on the in vitro model. And I'm going to show one example in the part two that we're applying this for disease modeling context. Where we use the neurospheroids as a powerful tool to identify first-in-class targets and compounds. The disease that we're going to be studying and discussing here is called Rett syndrome. So it's in the autism spectrum, although it's a little bit more severe than autism itself, and it's classified aside. It's an X-linked disorder with males being more severe effect. So it's a very difficult disease to model in vivo. So there is some in vivo models, some mouse models for this, but they not really recapitulate what you're seeing in the animal species. So this interspecies difference is really hard to be recapitulated in this particular model for this particular disorder. So that's where came the in vitro model. So it's, as I mentioned, the spectrum of phenotypes go from autism-like behavior, seizures, and loss of language, so it can be very severe. And most of the mutations are located in the MCP2 gene, so more than 95% of the case. So we took two different cell lines, one control line, that's a male line, is a parental control for the disease line, and another disease line that's is also a male, that's a nonsense mutation in MCP2 gene. So it's basically a knockout for that protein. and it's a very severe phenotype. The patient had a very severe phenotype or manifestation of the disease. Then one of the first things was to develop this in the 3D space, right? Develop these 3D spheroids that was mentioned, and see if there is if they're able to generate these spheroids in a very homogeneous way, because this is very important if you want to replace or use this in high throughput screenings. And here's a snapshot on the left, on the top for 3D4 well plate. So 3D4 well spheroids or mini brains that have on this for the Rett disease effect and for the controls. On the bottom here, you can have a bright field image of both of this as well, the Rett and control. And on the right here, you have a graph of the size, of the diameter of these spheroids over time. So you can see that even if you plate very homogeneous in the beginning, over time, they have a phenotype already just in the size. So there is a cellular phenotype that we're observing this over time on these spheroids. And this is very consistent. One thing that you can also do is start to play with these 3D models, these in vitro models, that now you can also match and mix. For example, as I mentioned, we started a male model that's basically there is all the cells contain the mutation, but this is X-linked disorder, and in the females you usually have a mosaicism in the brain. We have some neurons that express the mutant form, and some neurons that express the wild type form, the correct form. So we could now take these cells that we have in the lab and mix to match this mosaicism. So for example, create a mosaicism of 1-to-1, and now we have a phenotype that's usually in between the wild type and the Rett, the 100% Rett. And that's what's quite interesting to observe. So one thing that we can do as well with this model is, like, let's interrogate is there the correct cell composition there, right? Remember that one of the advantages of these in vitro models is that now you can have complex cultures in vitro, and this is really important for several disorders. One of the main advantages of the organism is because you have the complex tissue there, and there is many cases for many disorders that this complex tissue is really important to the manifestation of the phenotype. And so when you're talking about replacing in vivo model, you need to consider that physiology. You need to consider these complex models in vitro to be able to recapitulate the disease in a level that you can have a phenotype that's relevant to the disease in vivo. And here you can observe that you do have the correct cell types that are targeted. That's neurons and astrocytes, neurons in green by MET2 staining, and astrocytes in red by GFP staining in both control and Rett neurospheroids. You also observe diverse phenotype in Rett phenotype on the sculptures on molecular, cellular, and functional level. So these are going just for what was described in the literature and see if you can recapitulate those differences. And you did observe in the gene expression different expressed genes between Rett and wild type. Then we also observe a cellular phenotype that's the number of synaptic puncta between Rett and wild type reducing the Rett. And finally, this culminated with functional phenotype. That's basically a difference between Rett and wild type activity, electroactivity. So another way to measure the electroactivity, as I mentioned before, was through the calcium activity, right? So in this calcium, you can do in a high throughput scale because you can now do in 384 wells. And this is just showing two wells, so two different tracings for control and two tracings for Rett, regarding spontaneous neuronal activity, so spontaneous calcium activity here. So, and you can already see that visually you can see a difference there, right? One thing that's key when you have this biological data is like how you're going to extract the data. And one thing that we had to develop because there was not available was algorithms that are able to extract this data and quantify this difference on these two different phenotypes that we're observing between control and Rett. And finally, one thing that's key for progressing this as a high throughput option or high throughput screening platform is that it is reproducible, right? So between different batches of this differentiation, we're able to have the same phenotypes and phenotypes that are reproducible. So one of the key things that we have done is trying to combine this data science with this biological data to extract the right pictures, right? So we have this heat certification that's really important, and it's really a challenge in the field right now, because you need to have a huge data set of biological data to try to extract these pictures. And this can be easily achieved with these models that you now have in a high throughput fashion, that have the institute for form to have tons of the data points, biological data points that can now use to train these AI or algorithms that we're developing to make sure that they extract the right features. So in this example, we start with the calcium tracing features. We extract the features from these calcium weights that we observe, and then you can rank and summarize this data in a data plot of rescue. So we can quantify the rescue based on the controls and see how close it is after treatment you can reach the control levels of the activity. So for this particular study, we decided to use what's called the SMART library. So SMART is a collection of compounds that were vetted for Rett syndrome. It's composed of 296 compounds at 10 millimolars. And we did the primary screening at 1 micromolar. So you select one concentration, and the secondary screening at multiple concentrations. We used 4 replicates per dosage because this was easy enough. We use 4 replicates, we use 4 plates. You can achieve all of this. And we did a chronic treatment for 4 weeks. These are patients that's going to be for treatment for life. So remember that you want to mimic the correct physiology here. And these patients, they're going to have the mutation, they're going to have this density on the cells. So you need to make sure that the treatment is long enough to correct what needs to be correct and don't cause any side effects. So I'm going to show 3 different examples of what's expected and what you can find. One of the examples here, the first example is when you don't have, you treat with compounds, dose dependent, and you don't have any rescue. Moreover, at high concentration, start to have some toxicity. So the system, the beauty of this kind of systems that's able to even identify toxicity at the high dosage. So what you can see here is as you increase the concentration of the compound, and there is 4 replicates of the treated rat. At some point, you start to have no signal anymore. So there's a functional toxicity there that can lead to side effects in patients as well. So we need to be this system able to capture that and extract this information as well. You can also extract cell health, for example, ATP contents of the cells, and see if there is any decay on the cell health after the treatment with the high dosage. And this can be hand-to-hand with the biological data that we have, the functional data that we have here. There is this example that we have a good rescue. So as you increase the concentration, we have a dose-dependent rescue on the cells, and the signal start to become better and better over the concentrations, and there is no cellular decay. And finally, there is another example that you do have a rescue at certain concentration, but as you increase the concentration, we start to have side effects. And these are very common. Like if there's some compounds that they can have benefits on the disease, but at the same time, as you increase the concentration, you can have also side effects on this. And this system, functional assays like this one, that high focus scale are able to capture these potential side effects. And remember that this is a human tissue that sometimes can be missed on the in vitro model. So it's really nice to go hand to hand, understand this model with the in vivo model that we have, with the animal models, to see if this also can be observed in the in vivo models. So finally, what you do with all this biological data that extract, you do then hit identification and rank ordering. Again, we're developing new algorithms to do this, and there's a lot of research being done on this. That now that you have this biological data, so combine this biological data that's in a high throughput, so you have large library of this data, with advanced analytics to stratify the most effective compounds and targets. And now we start to rank the ones that have good rescue and no toxicity, no functional toxicity or no cellular toxicity, with compounds that would move forward. And this can already start to filter more compounds before they go to animal use or before they go to clinical research. So here's one example of what we found. Is basically that phenotypic recovery by target. So you can see that there are several targets that was composed on this microlibrary. You can see here the ones on the right is just the legend of what's considered rescue or not. So everything that was green or blue was considered some recover of this, and red is the one that not only didn't recover but had a side effect, so the decrease in the activity that could affect the patient. So we exclude those compounds and just focus on the ones that are in green and blue. And you can see there, that's based on the mechanism of action, there is a diverse, some that work, some that didn't work. But when you give a deep dive in particular mechanisms of actions, for example, the cholinergic system, then you start to observe something that's very interesting. If you go focus only the cholinergic system, then you have agonists and antagonists that have a mix of work and didn't work. And in fact, most of them didn't work. But when you focus on the acetylcholinesterase inhibitors, there is from 7 compounds, 6 have a recover, some level of recovery, 5 with a good, very good recovery. So this is already indication that this class of compounds for this disease is really important a class that can be a target on this. So when we move forward with this hit validation that now you can do in this platform as well, you start to observe that it's in a dose-dependent manner. You have this rescue. So you will move four compounds for this hit validation, and all these 4 compounds in the acetylcholinesterase inhibitor class, they were able to rescue the phenotype of Rett. And we did apply this for synaptic puncta and the MEA electrophysiology, and they also observed this rescue. So this is a very powerful indication that this primary screening that you can do at high throughput scale now can move and be validated through secondary assays to move towards preclinical research. So our compounds will confirm the rescue, and no compounds induce control line side effects. Or in other words, we also test this compound concentration in control lines to make sure that there is no side effect, functional or cell health side effects observed in this, and you didn't observe any of this with these compounds. So, of course, this is still hard because Rett syndrome is not a disorder that there is a compound that's available for treatment. So how you translate this research? So one thing that we found is that there is a clinical compound that's in phase 3 of clinical trial, that's NBX-273, and this is a sigma-1 receptor agonist. So we decided to use a similar compound also in this class, that's also sigma-1 receptor agonist, to see if you can do a clinical translation of this compound based on what is most promising right now for Rett syndrome. And we did observe a dose-dependent rescue of this Rett using this class of compounds. We used 2 compounds in this class, both had some level of rescue. So this is a very good indication that compounds of sigma-1 receptor agonists, it can move forward. So we were, based on the data that's coming out of the clinical trial with NBX, if it's positive, it is a good indication that these models can be used to identify compounds to move in preclinical research as well. So finally, just to summarize this part 2, human iPSC-derived neurospheroids, they can successfully capture key molecular, cellular, and functional phenotypes in vitro. We do observe a highly homogeneous functional phenotype on Rett spheroids. Using this platform, we identified 27 compounds on the primary screen is able to rescue the phenotype of the Rett phenotype that we have. And the hits that we select to move forward with these compounds, they did show a recovery of Rett phenotype secondary screens. And for secondaries, you use the dose response on flipper, synaptic puncta, and MEA electrophysiology. So this is a very promising platform to move compounds forward. So with that, I would like to first acknowledge the people that were involved in this work, and also the organizers for the event that allows this, we present the work here. So thank you very much, everyone, for everything.